Thanks, y'all. My name is Arietta, and I am a PhD candidate at the Yale School of Forestry with David Skelly. And I'm trying to answer the question, can adaptation counteract environmental change through the evolution of intrinsic rates? And I work specifically with these cute little buggers, which is a system of wood frogs here in North America. And to kind of lay the foundations of this talk, I'm going to set up three predicates. So the first is that ecological systems will be subject to increasing environmental change over the coming decades and centuries, which is probably pretty controversial. Second is that we should want to predict how populations will adapt to environmental change from the perspective of conservation biology in terms of management, and from the perspective of evolutionary biology, in that we can't really say that we understand evolution until we can predict evolution to some extent. And then third, that a change in environmental conditions in space is analogous to a change in environmental conditions over time. So I'll break that out just a little bit more. So for any variation in traits that we see across populations or within populations, we can decompose that down into the variation that's due to genetic components, the variation in those traits that's due to strictly environmental influence, and then their interaction and the covariance of genotypes and environment over space or over time. So specifically, I work with wood frogs. So if we take the environmental gradient of pond temperatures, so you'll see this is a pretty aggressive pointer angle, but we'll see if I can make it work. We're going to be talking about wood frogs, so if we look at dark, cold ponds versus warm, bright ponds, we can put that as our environmental variable. And the phenotype that I'm interested in is developmental rates. So across this environmental gradient, wood frogs are ectotherms, so in general, when they are in colder environments, they develop more slowly than when they're in warm environments, due strictly to environmental influence. However, assuming that it's not always advantageous to develop really slowly in cold environments or really fast in warm environments, we can expect over this gradient to see some local adaptation in the genetic component that countervails this environmental influence, so that we'll see something towards an average realized phenotype across the range. In 2001, my advisor Dave Skelly did a common garden that showed that this happens with our wood frogs. So in the wild, embryos that are in cold ponds, cold dark ponds, tend to develop really slowly. Embryos that are in really warm ponds tend to develop very quickly. But when we bring those into a common garden environment, we rear them at the same temperature, so taking out the variation due to environmental influences, what we see is that embryos from darker ponds of provenance actually have increased developmental rates compared to those from warmer ponds of provenance. So there is this counter-gradient variation. My question is, can we use this counter-gradient variation to predict contemporary evolution? So 2001 was 18 years ago now. This is 2018 when I did my project. And I decided that we could use this to kind of use a space for time conversion. So if we want to predict how temperatures have changed over time, we can make that same inference for space. So if we think that temperatures have increased, so the environmental influence has increased, we might predict that local adaptation will drive intrinsic rates to decrease over time, just as we've seen across space. And that's what I actually put on there, didn't even need a pointer for. So I replicated Dave's study. We chose 13 ponds. From each of those 13 ponds, I grabbed 6 clutches within 24 hours of oviposition, so 24 hours after they were laid. And then from each of those clutches, I excised a dozen individual embryos and raised them individually in this common garden at 2 different temperatures. So we had about 888 data points. And one thing that I'll point out about the ponds, 9 of them were overlapping with those from Dave's 2001 data set, is that they are only, at maximum, they're about 4.5 kilometers apart. And the 95% genetic dispersal kernel for wood frogs is somewhere in between probably 1 and 2 kilometers. So this is extremely microgeographic. Almost all of the dispersal kernels of these ponds overlap. And what we see, just looking at the environment over this time, what I'm plotting here is the difference for each of the ponds, the difference between 2018 and 2001 in temperature, is that, as we might expect from climate change, the ponds have gotten warmer. This is spring pond temperature, by about 2.3 degrees warmer over the 17-year period. But there's a lot of variability in the ponds. And then when I look at the results from the 2 common gardens and look at the difference in developmental rates, I see that they are on average about 15% faster. Intrinsic development rate is about 15% faster, which is the opposite of what I just told you I was expecting from a few slides ago. But again, there's a lot of variation. And to understand why there's this, what I would think is a counterintuitive result, we have to look a little bit into the natural history of wood frogs. So wood frogs are one of the most cosmopolitan amphibian species in North America. They're the only frog found north of the Arctic Circle. They're kind of famous because they can freeze almost entirely solid during the winter and then reanimate in the spring. And that's because they breed really, really, really early in the spring. So this is a wood frog breeding pond while they're breeding. This is one of our males who's literally sitting on top of a frozen pond waiting for it to melt out from underneath of him. And he was joined by probably 4 dozen other frogs that were all sitting on top of this pond. And the reason that they do this is that they have this evolutionary impulse to get into the ponds really early because their ponds dry out throughout the summer. So they need to get in as early as possible, develop quickly enough so that they can metamorph before the pond dries out. Otherwise, they're tadpole-shaped raisins at the end of summer. And we also need to look at how the environment has changed in the intervening 17 years since that first study. And for one, even though we know that average temperatures have increased annually, that's not homogeneous across the season. So early spring temperatures in our region have actually gotten a little bit colder, and that's precipitated in a slowed or delayed snowmelt. What that means for wood frogs is that the initial period of their developmental period has been kind of frame shifted back. So oviposition happens about 2 days later than it used to. So we've seen this kind of phenological shift. We also know that the summertime air temperature later in summer has been increasing, so there's more evaporation. And we also know that canopies have closed in by about 7%, and that green up comes about 2 days earlier. So there's more leaves, and the leaves come on earlier, which through evapotranspiration sucks the water out of the ponds much faster. So the net result is that we see this contraction on both sides of wood frog development by snowmelt and by reduction in hydro period, so that if they do nothing, we will have tadpole-shaped raisins in every population. But what we've seen in reality is that they have contracted their development period by increasing their developmental rates. And to look at this, I looked at all of our specimens. We've been studying these things for a long time, which means we've collected a lot of tadpoles, about 17,000 tadpoles, and plotted their developmental stage against the day that we collected them, the day of year that we collected them. And what you'll see is that over time, these slopes are the fixed effects from a mixed effects model, that they are increasing in slope over time. So developmental rates are increasing over time, the realized phenotype that is. And this is due, all due primarily to this hydro period effect, less so than just the temperature effect or the environmental effect on that, on the phenotype. So interpreting this back in the context of this temporal change that we saw in temperature and this temporal change in intrinsic development, if I plot those two against each other and use the standardized, so the mean-centered development rate in each year, so kind of discounting that directional change that we saw across all populations, what we see is that those populations that had the greatest increase in pond temperature actually show a decrease in developmental rates, as we would predict from the countergradient information from 2001. And that those ponds that saw almost no change in temperature, or even cooled a little bit, have an increased developmental rate, exactly what we would expect, so countergradient to that variation. Countergradient variation with respect to the environmental variable. So the conclusion here is that countergradient variation can help us predict adaptation into the future, but we have to interpret it in the context of attending shifts in natural history, which we may not be able to predict. Another quick point is that this study shows that adaptation can be really rapid and very microgeographic. So 17 years is maybe at maximum 8 or 9 wood frog generations, and we're seeing these huge shifts. And then also a 5-kilometer, basically, range in ponds is almost nothing compared to this giant range of the wood frogs. And I think maybe most important is that it's really important to replicate these historical field studies. And I would argue that probably the biggest hurdle here is convincing PIs that the work that they did at the beginning of their career should now be considered historical. The the obstacle of the existential crisis that you have to get over to do this. So with that, I'd like to thank, I guess first and foremost, my advisor Dave for getting over the existential crisis, and then all of the folks in my lab who have contributed some of their legacy data, and my funders. If you want to find out more about all this, there's my website and my email, and I'm happy to take questions, I think, with
I think you said at the beginning that the ponds had warmed up by 2.3 degrees.
the spring temperature, so that's actually just May.
So if that's due to climate change, why do you think ponds are warming so much faster than terrestrial water?
I think, well, so when you look at just air temperatures in our region, the air temperatures have increased by about 1.6 degrees in that time period. So, which is more than what our regional average. So some of it is just microclimate. But then pond temperatures, and I've been going back there and looking at what actually drives pond temperatures. We've had hobo loggers in these things for basically over the past 20 years. We have pretty good data, but it's a combination of their where the water is feeding into these ponds from. It might be draining from snowpacks farther away. There's a canopy effect, and there's net radiation gain. There's just lots of things that are going into the microclimate of these individual ponds. Yeah.
So I'm on board with adaptation happens fast, but presumably it can only do so based on there being continuous genetic variance, right? So if the historic study was one of these split sit studies, so was yours. Can you or have you assessed variability change for the development rate?
We haven't.
Between then and now?
We haven't, but that is part of what we're hoping to do going forward. I think that sound means that I finished really early, so if you have any other questions.
Really interesting talk. I did want to point out, as a historical person, that soon your research will be historical as well.
Point taken.
It happens fast.
And just what about size then? If they're developing faster, are they coming out at a smaller size?
That I'm also looking at. And we don't necessarily have that in common gardens, so we can't decompose into the environmental influence and the genetic influence. But I can look at that with the museum specimens that we have to some extent. And it does seem like there are some trends, but I don't really know enough about what I'm seeing to say much. I will also say that on the other end, and it's part of this research that I haven't talked about, is does seem to be some potentially adaptive maternal provisioning as far as embryo size. So if the embryo starts out bigger, it develops faster. And the size of the embryo correlates with the canopy gradient. Yes.
So the canopy gradient variation that you saw was for growth?
Development.
Development. But I might have missed this. Did you see evolution of the slope of that reaction more through time?
Yeah.
Okay.
Mm-hmm. But it's very pond specific.
Yeah, the natural history part of it is really interesting, and I'm thinking about it. It seems quite specific to this system, but maybe, would you think that the same sort of effect of earlier leafing out, higher canopy might generalize to forests in the Northeast, or?
I don't know. This is about the only spring advancement in phenology of any of the